I'm Kong Anisa from Neurophysiology Laboratory. In our laboratory, we study to investigate neuromechanisms of human homeostasis. The brain uses various regulatory systems to adapt environmental conditions and try to maintain human homeostasis. For instance, blood circulation and respiratory movement are regulated by the neural system. So now we focus on the neural systems to regulate the blood circulation and respiratory movement. Essential organs to regulate the blood circulations are heart and vessels. The heart and vessels works as a pump and pathway for the blood circulation. So heart and vessels are called as cardiovascular systems. The functions of cardiovascular systems are regulated by neural systems. So the brain can regulate the cardiovascular functions and blood circulation. In other words, the central nervous system regulates the blood circulation. From here, I'd like to introduce research topics of the Neurophysiology Laboratory. Now we are focusing on the stress-related circulatory regulation by the central nervous system. Usually we are facing various types of stresses. Stress events usually change the behavior. However, in our body, circulatory system also try to adapt the stress conditions. For instance, with facing the stress events, we sometimes try to run away from the stress. In this situation, the skeletal muscle requires large amount of blood, and the body tries to cause the vasodilation at the skeletal muscle. This kind of vasomotion is modulated by central nervous system. So stress events change not only the behavior but also the blood circulation that is mediated by central nervous system. In order to investigate the neural mechanism of the circulatory responses to stress, we focused on the brain region lateral habenula. Lateral habenula is a popular brain region that it can modulate the behavior and the condition of the stress events. So we hypothesized that the lateral habenula also regulates the cardiovascular functions. So as a result, activation of the lateral habenula changed the cardiovascular parameters like heart rate and blood pressure. So this indicated that the lateral habenula modulates not only the behavior but also the cardiovascular systems that is mediated by autonomic nervous system. Additionally, we also found that this lateral habenula and autonomic nervous network is mediated by central nervous system. After this finding, we reported this finding into the international journal and released into the press. Now we are also focusing on the brain ischemia related circulatory regulation by the central nervous system. On this slide I showed the picture of giraffe and human. As you can see, the giraffe has a quite long neck, so to deliver the enough amount of blood into the brain, giraffe have to have the high pressure around the heart. Otherwise, the brain cannot receive enough amount of blood from the heart. On the other hand, humans do not have such a long neck, so blood pressure around the heart and brain is similar in the human being. However, potentially, human also has a similar blood pressure regulatory system with giraffe. In the human body, when the blood circulation of the brain decreases, body tries to increase the systemic pressure and the brain receives enough amount of blood from the body. So this reflex is popular as Cushing reflex. However, even now, the question is arises as to how the brain ischemia is detected. So we tried to investigate the system to sense the blood circulation of the brain. As a result, we found that special neurons in the brain can detect the dropping of blood circulation of the brain, and also these neurons can elevate the systemic pressure that is mediated by activation of the autonomic nervous system. As a result, elevation of the systemic pressure can improve the blood circulation of the brain. Okay, so what is the importance of this finding? The keyword is hypertension. It has been reported that in the hypertensive patient, blood circulation of the brain is lower than the normal. And also in the hypertensive patient, cardiovascular sympathetic nerve activity is overactivated. The previous system is important to maintain the blood circulation of the brain. However, in the hypertensive patient, blood circulation of the brain is continuously lower than the normal. So, this dropping of the blood circulation of the brain can activate the special neurons that can detect the blood circulation of the brain. Activation of the special neurons can activate the cardiovascular sympathetic nerve activity continuously and can cause the hypertension. Therefore, more investigation about the details of this system is quite useful to find the new therapeutic target for the hypertensive patients. In the Neurophysiology laboratory, we are also focusing on the respiratory regulation by the central nervous system. Respiratory movement can take oxygen into the body and can release the carbon dioxide from the body. So this is quite important system to maintain the human homeostasis. Respiratory movement is generated by a respiratory center of the brain. The respiratory center is composed of various types of neurons, and these various types of neurons make very complicated neural circuit. and the respiratory center. This complicated neural network can generate respiratory pattern and output. However, the details of systems to generate a respiratory pattern output and modulation of a respiratory output are still unclear. So now we are focusing on the respiratory regulation by the central nervous system. Here I'd like to introduce an example that is showing the hydrogen sulfide related respiratory regulation by the central nervous system. Hydrogen sulfide is popular as a toxic gas. However, it has been reported that hydrogen sulfide is also generated in our body and has some physiological functions. In the brain, also some physiological functions are reported. However, in the respiratory center, nobody knows about the function of hydrogen sulfide to generate a respiratory pattern. So, we tried to investigate the function of hydrogen sulfide in the respiratory center. As a result, we reported that hydrogen sulfide was really essential to maintain a normal breathing. So in other words, the hydrogen sulfide is generated in our brain, and this endogenous hydrogen sulfide is really important to generate the normal respiration. This finding was the first report that is reporting the physiological function of hydrogen sulfide to generate the respiratory activity. So we published the paper into the international journal, and also we had a press release like this. The neural regulation of a respiratory movement is essential for human homeostasis. Therefore, impairment of this system causes respiratory dysregulation. For instance, people believe that the sudden infant death syndrome is caused by respiratory impairment in sleep. However, the detail of this system is still unclear. So we believe that investigation of the neural mechanism to generate respiration in the brain is really useful to find the new strategy to approach into the neural disorder or dysregulation of respiratory system. In this presentation, I briefly introduced our research topics in the Neurophysiology laboratory. If you are interested in our research activities, please visit our website or Twitter to find the latest information on our research activities. If you are interested in the Neurophysiology laboratory, please contact us with using this contact information. Thank you for your attention.